So, for those of you who don't know about respiratory syncytial virus, or RSV, it's the commonest cause of severe respiratory tract illness and hospitalization of infants in the whole world. So this is a global disease, 30 million cases annually and nearly over 3 million admissions. Just under 200,000 deaths thought to be attributable to RSV, and the vast majority of these deaths occur in resource-poor countries. Now, in the more developed nations, we know RSV for its role in nosocomial transmission and causing outbreaks on pediatric wards. And also there's data to suggest that RSV has a role in causing wheezing in later life and may even predispose children to developing asthma. Now, this hasn't been definitively established, but there are recent data to suggest that if you can prevent RSV in the first 12 months of life, you can actually reduce subsequent wheezing. So there is increasing body of evidence to suggest this virus does have a role in later wheezing. There's no licensed effective antiviral for RSV at all, and despite a lot of efforts, we still haven't created a vaccine. And the focus of my work is trying to address perhaps why that is. So RSV is not just a disease of children. There's a substantial morbidity and also mortality in adults, particularly the elderly, and particularly those with comorbid conditions such as chronic obstructive airway disease and other forms of chronic heart disease. It's useful to draw the comparison between RSV and influenza. Influenza, as you all know, being probably the other major cause of respiratory morbidity and mortality in the world. They're in different virus families, but they have some similarities and differences. Now, in terms of immunity to influenza, you probably all know about hemagglutinin and neuraminidase. Those are the two major antigens, and they're both highly variable. So you're familiar with the phenomenon of antigenic drift and antigenic shift, whereby influenza can vary from season to season and occasionally more dramatically. Now, RSV does undergo seasonal variation, but not to anything like the same extent. The F protein, the fusion protein, is actually highly conserved among all strains of RSV, and it's possible to make neutralizing antibodies that can actually neutralize all known strains. G, the other major glycoprotein, is more variable, but nothing to the extent of influenza. So it seems that antigenic variation is not a major role that RSV persists among humans. In terms of flu, you can get immunity to specific strains. If you've had one strain of flu, you can't be reinfected with the same strain. However, with RSV, it's quite possible, following a natural infection, to reinfect people time and time again with an identical virus. All right? So it's not due to variations in the virus. It's something to do with failure of immunity. We have flu vaccines. We have several flu vaccines. We have inactivated ones and also attenuated vaccines. Now, neither of these are especially effective, and as you all know, we have to have revaccination annually with flu, and sometimes there's mismatch between the circulating strains and the vaccine, as happened in the previous season last year, and sometimes the vaccine efficacy can be very, very small. Now, there's no licensed vaccine for RSV. Now, I'm going to try to talk about some of the reasons why this is. Certainly, RSV itself doesn't seem to be particularly immunogenic. We don't really understand what constitutes immunity to RSV. In fact, there's no such thing as an individual who's completely immune, at least not as we know. And there's also safety concerns following a trial of a formal and inactivated vaccine in the 1960s that mean that vaccine development has been very problematic. Now, just talking about correlates of protection, so this is a very interesting study done a number of years ago by Caroline Hall, who went on to show that you could reinfect people again and again with the same strains of RSV. So what she did was take 15 people who'd had a natural infection and follow them for just over 2 years, and then re-challenge them experimentally with a laboratory strain, an identical strain of virus. And what she showed was that even after repeated challenge, you saw continued susceptibility. So people developed symptomatic infections even after their 5th or 6th challenge with the same virus. All right, and although they had high levels of antibodies at their first challenge, and those antibodies did increment in the initial re-challenges, what they did observe was a plateau in the boosting of antibody responses, and the fact that you can't seem to raise the antibody levels high enough in order to produce total immunity. Now, in terms of what correlates of protection we do know about, well, serum neutralizing antibody is the best studied, and this study done by Pedro Piedra in 2003 looked at trying to define a correlate of protection for RSV, and they noticed that children, as they got older, were less likely to be hospitalized, and there's lots of reasons for that, not least because older children are more robust than younger children, but also they have higher levels of antibodies in their serum, and so they showed the relationship between age, antibody level, and hospitalization risk, and went on to show that, those with the higher levels of antibodies, irrespective of age, were less likely to be hospitalized than those with the lower antibodies. But the relationship is far from complete, and as you can see here, although the graph reaches significance, there doesn't seem to be a level above which you get immunity. So what do we know about mechanisms of protection? Well, I've talked a bit about antibodies, so serum neutralization antibody is the best study, but it's clearly an incomplete correlate of protection, and given the fact that, RSV is an illness that's really confined to the respiratory tract, it's not at all surprising in some ways that serum antibody is not going to act to protect from infection in a great way. Palivizumab is a monoclonal antibody made in mice. It's a humanized mouse monoclonal, and it acts against the fusion protein. And we know that palivizumab does work. When used properly, it can reduce hospitalization by nearly 80%. But what it doesn't do is prevent infection entirely. It seems to protect against lower respiratory tract disease, against bronchiolitis, the more severe complications of RSV, but it doesn't prevent upper respiratory tract infection. And the other thing that it doesn't do is act as a therapeutic. You can't give monoclonal antibodies to people with established disease. It doesn't make any difference. It doesn't help them get any better. So what about cell-mediated immunity? Well, T cells have generally been studied in mouse models of RSV. Now, I think there's been some great experimental work that's come out of that, but it's very clear now that the disease that you can produce in mice is not the same as the disease that you can produce in humans, and in some ways that isn't that surprising. However, what you can show is that T cells are very important for clearing virus. Well, I think that's what we would expect, and they shorten the duration of viral shedding, but they can also actually enhance the disease. So if you perform studies where you deplete T cells, you can actually reduce apparent symptoms in the mouse model, and if you perform adoptive transfer of RSV-specific T cells, you can actually enhance disease. So the role for T cells in protection is not clear, and we haven't really been able to study them in human beings simply because it's actually very, very difficult to do this. So what about vaccine-enhanced disease? Well, I mentioned that a formalin inactivated vaccination was trialed in the 1960s after the success of formalin inactivated polio vaccines. Now, unfortunately, children who were vaccinated with this particular vaccine in a subsequent RSV epidemic that occurred about 4 years later, they had a much higher incidence of hospitalization for pneumonia than children who'd received an unrelated vaccine. And unfortunately, there were 2 deaths in this cohort from severe RSV pneumonia. And we don't really understand why this is. There's been a lot of research into why this vaccine caused such severe disease, and it's thought to be related to dysregulated immune response. I mean, the vaccine produced antibody levels. It seemed to be safe in guinea pigs when it was tried, but subsequently in children, it looks like the antibodies that were produced probably waned over time, were poorly neutralizing, and certainly this vaccine did not prime for long-lasting protective immunity. And this experience has really blighted vaccine research into RSV because, of course, what everyone is very, very worried about is producing a vaccine that is going to actually produce enhanced disease. So that's really stifled progress. However, there is a lot of research going on into RSV vaccination, so this is not a neglected disease by any means. However, it is still a great unmet health need. You can see most of the trial vaccines here are very much in the preclinical stages. So these are, really being evaluated in animal models, in vitro, for instance, and a few have made it into phase 1 studies in volunteers, and even fewer candidates have got further into phase 2 and phase 3. The 3 broad approaches that are being used here are either trying to protect children. Now, this might be a vaccine that you can give to very young infants, and that in itself is a huge logistical challenge. But the other strategy that looks quite promising, as one of our colleagues alluded to earlier on, is maternal vaccination. So we know that very, very young infants have high levels of antibodies acquired from the mother, and they appear not to suffer severe disease from RSV. So if you can somehow boost the antibodies in late pregnancy, you may offer some protection to newborns that may last as long as 3 or 6 months. The other group that's being targeted are, as I mentioned, elderly people. So elderly people suffer quite considerable burden of disease from RSV, and obviously a cohort that's perhaps slightly easier to do a vaccine study in than is young children. I mean, all the vaccine candidates have got far at all have been those that are based on the F protein, the fusion protein of RSV, these are all being trialled primarily in adults at the moment. So what can we do with experimental infection studies then? Well, they have a number of advantages over conventional approaches. Obviously, they're done in human beings, which is a major advantage when you haven't got a good animal model, such as in RSV. You have a defined inoculum, so you know what your subjects are getting. So when you're studying a natural infection, there could be viral variation, there could be co-infections and other things that may affect what you're seeing. We can select our subjects, and that can either be because you want healthy people with no comorbidities, or perhaps you want very, very defined comorbidities to study the effect of whatever you're doing in a very defined population. We can assess the pre-infection immune status of our subjects. And this is a major advantage when compared to studying natural infection. By the time people come to hospital, they've often been sick for several days, and whatever events may have led to them getting so ill and being hospitalized have often passed, and you can't measure their effects. You can obviously sample people intensively when you do an experimental study from baseline and then daily, or even potentially several times in a day, to get a real high-resolution, longitudinal picture of what's going on. However, there's a number of disadvantages of this approach. Obviously, the whole ethics approach in that, when you're going to make someone sick deliberately, you have to have sufficient scientific justification for doing so. But not only that, there is a limit to what you can expose people to, irrespective of whether or not they agree to be involved. These studies are very, very expensive. You obviously have to pay your volunteers for their inconvenience and their time. You need to obtain challenge agents, which are in very much limited supply and quite expensive to produce. There's not a big market for biotech companies to sell these, so the unit cost of challenge viruses is very, very high. And also, you're only looking to produce mild or moderate disease, so that only potentially allows you to study a limited spectrum of disease phenotypes. So how can experimental infection studies help with vaccine studies? Well, we know that vaccine development is very, very costly, and those costs are rising. Failure at the late stages can mean that, years and years of work and years of expenditure have been lost. And the trials that you need to get licensure these days need to be extremely large. So very often, as we've heard earlier today, it's difficult to get companies interested in doing these trials simply because their final market is either uncertain or too small. Challenge studies can help here because they can act as a proof of principle. You have a higher infection rate than perhaps you do in a natural study, so you have more potential endpoints to look for, and you need smaller sample sizes. And they also permit you to do exploratory research into pathogenesis, what I'm interested in. However, you can't do these in truly at-risk individuals. So, whilst you might think about conducting, a challenge study in the elderly, healthy elderly, or perhaps in mild asthmatics, what you can't do is do this in infants, and what you can't do is do this in the immune-suppressed. And these two groups are particularly vulnerable to RSV infections. And, this is optimal conditions as best as you can expect for a human study. So they don't necessarily recapitulate all the aspects of natural infection. So what do? Well, we look at healthy adults. Our age criteria are pretty broad: no regular medications, allergies, and no colds. We give them an intranasal inoculation. We use 10,000 plaque-forming units just to give an idea of what that means. The minimum infectious dose is thought to be probably around 500 to 1,000. So we're giving a high dose here, the idea being that we want an infection. They're quarantined for 10 days. That's quite logistically interesting. And they can undergo daily sampling from blood, from the nose. And we also managed to get some of our subjects to undergo bronchoscopies, both at baseline, during infection, and at convalescence. So great credit to them. They're followed up after the study for up to 6 months, looking at their serology and their peripheral blood cellular responses. So here's how we do inoculation. So, this is the past here. As you can see, it hasn't changed a great deal here. You may recognize this figure here wearing PPE. Seems slightly over the top for a virus that's supposed to cause minimal disease. I did have a hard time explaining to my subjects that there was nothing to worry about at all. So we've managed to inoculate 61 people, and one of the surprising facts is actually so many people don't get infected at all. So 44% no infection, so no virus detectable at any point after the second day by PCR or culture. Now this is surprising because you're sticking 10 times the minimum infectious dose directly into the nose of a person. So if that can't infect them, you think, well, they must be pretty robust. And it also makes you wonder how on earth do natural infections really happen. I think what the actual explanation is that adults are quite resistant to RSV in general, having had it many times in their lives. Just over half of our subjects do develop an infection, and 2/3 of those get a typical cold lasting approximately 1 week. Interestingly, 1/3 of people who can demonstrate virus by PCR don't get any symptoms at all, or get minimal symptoms that don't constitute a cold. And that group is quite interesting, although I'm not going to talk about them today. Symptoms that you get correlate quite closely with viral load, both in terms of the kinetics of onset and also in their overall magnitude. Now that may be because when you've got a runny nose, it's much easier to detect the virus, or it might be that actual virus infection is contributing to symptoms. I think those two are quite difficult to disentangle. The symptoms are primarily upper respiratory tract, so nasal congestion, stuffiness, things that we may recognize as a cold. Systemic symptoms and lower respiratory tract symptoms, extremely uncommon in adults who are healthy. So what about protection? So the focus of my work really was to try to study correlates of protection. And so we started with serum neutralizing antibody because this is the most well studied and it's what's in all the literature on RSV about it being protective. Now we really didn't find any relationship at all. I mean, on the whole, those who are infected might have lower median titers, but I mean, the graph is completely unconvincing. Certainly, there are people who have very low titers in their blood for whatever reason. Maybe this is because they don't have any exposure to children, where I think RSV circulates in much greater numbers, or maybe they just haven't been here in the winter time when RSV is circulating in this country, and they're very vulnerable, and some people have selected those people out to do infection challenge studies with the aim to create more colds so they get more endpoints, and that's quite useful if you're trying to test a therapeutic, but not if you're trying to understand mechanism of protection. Now these people here have high levels, but they're not consistently protected. I mean, there's no convincing difference between these people here and these people here in terms of your numbers of infection. So serum antibody doesn't really seem to correlate with protection at all in this group. So what about local antibodies? So, we thought, well, this is a mucosal infection, presumably, local IgA must have a role here. And in fact, it seems to. So the relationship between protection and nasal IgA is much stronger, much more convincing. You can show that as you have higher levels, you're less likely to be infected with a logistic regression model. But again, there's no threshold that you can see here where you don't get infections or you're consistently protected. The vast majority of people pre-infection were in this sort of middle zone where they had really a 50-50 chance of getting infected, and that's reflected in the infection rates that we finally observed. Very few people achieved this high level after they'd had a natural infection. So, what this really suggests is that any vaccine has got to improve upon the immunity that we generate through natural infection. And the other problem is that the antibodies that you generate are very short-lived. Now, this has been shown before in natural infection, but not in experimental infection. So, we observed sort of 2- to 4-fold rises in titers following a successful infection in our subjects. But by the time we followed those up, about 6 months, the titers had waned back to almost the same levels that they were pre-infection, almost identical levels in many cases, suggesting that, most adults by the time they reached their 20s, which was the sort of the age that we had in our studies, that their antibody immunity to RSV is really maxed out from natural infection, and you can't really improve upon it without something unconventional. So what about generation of antibodies? We tried to understand why this is happening. So what we did here is we took whole peripheral blood mononuclear cells during infection and did a B-cell ELISpot really to look for antibody-secreting cells. And what you can see is that the total numbers of cells and the RSV-specific antibody-secreting cells are really absent in the early stages of infection. They only really appear in the blood at day 10. where there's quite a profound peak, and then they're very short-lived. There's a few still hanging around at day 14, and then they're completely gone. But you do get robust induction of both IgG and IgA producing RSV-specific antibody-secreting cells. And there doesn't seem to be a difference between the relative amounts of IgG and IgA. Not necessarily surprising, given this is a mucosal challenge and very much a mucosal infection. And we showed that you could correlate the fold change in antibodies following infection with the frequencies of these antibody-secreting cells. So it seems generation of antibodies and generation of antibody-secreting cells in RSV seems to function as expected. So why don't these antibodies last? And is there a problem with antibody memory? So what we did here was we looked at memory B cells in the blood. And what you do is you get whole peripheral blood mononuclear cells, you stimulate them in vitro with mitogens to activate memory B cells, and then you go and perform the same sort of B cell ELISpot that I showed you before. And what you can see here is that baseline frequencies of RSV and RSV-F specific cells don't differ between those of an unrelated virus or a slightly related virus, measles. So obviously most of the population have had either measles natural infection or vaccine, so you would expect to find those cells at low numbers, and indeed you do. And there are similar numbers to the cells that are specific for RSV. Now this is IgG. I haven't shown IgA here, but we weren't able to detect IgA memory B cells at baseline, which is interesting. So coming on to day 28 post-infection, now what we see here is increments in the IgG secreting RSV-specific memory B cells, like you would expect. So these increment appropriately, fourfold on average for RSV and twofold on average for F. And obviously the measles-specific cells are not changing because these people haven't been exposed to measles virus. So we're showing this is a virus-specific phenomenon, and IgG increment seems to be appropriate and seems to be intact. But what about IgA then? So we said that IgA was the better correlate of protection. What's happening with the memory here? Now, as I said before, we weren't able to detect RSV-specific IgA memory B cells at baseline at all. And that might be because these cells are quite rare. Now, even after day 28, you saw that there was quite impressive induction of antibody responses and acutely generated antibody-secreting cells. But what we're not able to detect at all are IgA secreting RSV memory B cells, despite the robust response during infection. Now, is this an RSV-specific phenomenon, or is it just that these cells are so rare that the assays don't detect them? Well, we try to address that question by looking at people who recovered from pandemic influenza. So we're quite fortunate to have access to some stored samples from patients admitted to hospital during the flu pandemic in 09-10 and 10-11. And what we were able to show here is that numbers of IgG and IgA memory B cells at convalescence in these patients were comparable. So, in flu, it seems that, you do get robust antibody memory and you get strain-specific immunity. However, in RSV, you don't, and the defect really seems to be something to do with IgA memory, and certainly the IgA memory B cell compartment is not incrementing. As to why this is occurring, now we haven't actually addressed that yet, but I suspect it's something to do with innate immunity. So coming on to the conclusions about antibody-mediated protection, well, I've shown you that mucosal IgA protects against infection, at least partially. Serum IgG seems to prevent lower respiratory tract involvement in so much as adults don't seem to experience symptomatic lower respiratory tract disease, and that palivizumab seems to work to prevent lower respiratory tract disease and bronchiolitis. We've seen that the RSV-specific antibodies are poorly maintained, and I think this is due to a low frequency of IgA memory B cells and a defect here. And it may also be that there's a relative defect in long-lived plasma cell generation and contributing to poor maintenance of serum antibodies as well. So the big question is whatever putative vaccines that we're going to develop, will they actually be able to boost memory? And perhaps if they're mucosal vaccines, they may be able to address that. So I'm going to come on to cell-mediated immunity now. So this is really the work of my colleague, Chris Chu. So what happens when immune exclusion fails? So obviously IgA is unique in that it acts in the mucosal space. It can bind viruses and other pathogens and completely exclude them from the body. But what happens when you overcome this? Well, we know that with repeated infection, you get milder disease. Older children and adults don't really come to hospital with RSV unless they have comorbidities. In terms of cell-mediated immunity in animals, we know that T cells reduce viral load, but have also been shown to exacerbate symptoms. In natural infections in humans, we know that children or adults with T cell deficiencies have often a protracted course with respiratory virus infections and can suffer quite severe disease, including viral pneumonitis, when they encounter RSV. So T cells do seem to be important in protection from disease as well. So here we've got some data on the experimental infection looking in the lower airways. So as I said, we managed to convince our subjects to undergo bronchoscopy. So here you can see the appearance of the airways at baseline, and then there's day 7 of the infection and day 28. And what's very striking is that these people didn't have symptoms of lower respiratory tract disease at all. They're healthy. We didn't expect them to get any pathology in their lungs. But at day 7, I mean, the airways, are pink and friable, and even at day 28, they're clearly abnormal and inflamed. You're able to detect the virus by PCR in BAL and also bronchial brushings around day 7 and 10. It's largely gone by day 28. And also by immunohistochemistry, you can see here at baseline, and then here with RSV antigen is stained brown. It's quite widespread throughout the epithelium, and it's persistent even at day 28. I mean, this is very, very striking. This is a disease that's not thought to be a lower respiratory tract infection in adults, but clearly it is, but it's in the absence of symptoms. And we also went on to show that T cells are infiltrating the respiratory mucosa here. So you've got, again, the histology. The CD8 T cells are stained brown, and you can see they're infiltrating the mucosa here at day 7, and they're still persisting until day 28 here. Oops, sorry there. And in terms of the antigen-specific T cells and the phenotype of the T cells. So these displayed markers of activation. So CD38 and Ki67 are markers of activation in T cells. And in the blood, they were peaking at day 10, which was the same as you might note as the antibody secreting cell response. And also in the lung, but much more enriched in the lung here. So much higher frequencies in the lung where in the airway where the infection is taking place than in the peripheral blood. And when it came to the antigen-specific cells, so we were able to make some RSV tetramers to stain the antigen-specific T cells. A similar sort of picture is seen. So much lower frequency, obviously. Antigen-specific T cells are rare. But again, peaking at day 10 in the blood, but slightly different picture seen in the lung now. So, I mean, again, they started to rise at day 10, but the numbers continued to increase into convalescence, and actually the highest numbers were detectable at convalescence in some subjects, up to 3 to 4% of CD8 T cells were RSV specific. I mean, this is quite a striking finding in that, even when they've recovered from their illness, you're getting high numbers of antigen-specific T cells still in the lung here. And what does this represent? So we went on to try to examine the phenotype of these T cells, and it seems that they express the markers CD69 and CD103, for those of you not familiar with T cells. These are the canonical markers of resident memory T cells. So these are cells that are not thought to circulate throughout the body and into the blood. They're thought to be resident in the mucosa and acting as somehow a ready armament to defend against infections at mucosal sites. And what we were able to see here was that in the blood, I mean, the phenotype of these cells was primarily the CD103 only, but in the BAL, so in the airway, they were double positive cells. So these are resident memory T cells in the lungs. We went on to look about whether these cells, given the fact they're in the lungs, seemingly all the time, can they prevent against infection in the way that antibodies seem to be able to? And the answer is they don't seem to be able to. So for those subjects we were able to measure this, we didn't see a difference between, in terms of frequencies of these RSV-specific T cells in the lung at baseline, between those who subsequently became infected and those who remained uninfected following challenge. But what we did see was that these numbers correlated quite well inversely with symptoms in terms of cumulative symptom scores for the course of infection, and also peak viral load. So it seems that these resident memory T cells are somehow able to control viral infection quite effectively and reduce disease severity. So just concluding about what we found out about T cells, so it looks like the T cells in the blood really poorly reflect what you're seeing in the lung, and if you want to examine this, you really have to go into the airways. It's possible that T cells in the blood are an indirect correlate of protection, but they're very, very rare, and they're very, very short-lived. But they are very highly enriched in the airway, and we've shown that these reduce severity of RSV disease, and that these cells are resident memory T-cell phenotype, so they're staying in the lungs, and they accumulate, and after actually in early convalescence, they're at their most abundant, perhaps maintaining an effective immune barrier to reinfection during an epidemic, in a similar way that the increments you see in convalescent antibodies may do. We didn't see any evidence of increased disease severity due to CD8 T cells. Now, as to why that is unclear. It may be that in the lung, being that the environment that it is, there's a lot of regulation to prevent immune pathology. That would make a lot of sense, but it's not entirely clear why. And it may be that induction of these T cells can protect against disease when antibody is overcome. So what are the implications for development of vaccines? Well, in terms of where we might interrupt viral infection and disease in RSV, well, we know that antibody can obviously prevent establishment of infection. Now, what I didn't show you, but what we have shown is that it has no effect on subsequent disease if it's overcome. So in those people who get an infection, their pre-existing antibody levels have no protective effect on the extent of the disease they get in terms of symptoms or in terms of duration or peak of viral shedding. However, it does seem that T cells have a role here for controlling viral replication and for clearing virus and for reducing severity of disease in terms of symptom scores and viral shedding. So probably do have a very important role to play in terms of resolution of infection and in convalescence. The role for serum antibody in terms of controlling viral replication or mucosal antibody even in controlling viral replication once disease is established is unclear and probably has very little part to play. So in terms of what we know about for vaccines, well, mucosal antibody, I think, should be the aim of any putative RSV vaccine. I think the F protein is the most obvious target. It's a protective antigen that is subject to the least variation between RSV strains. However, whatever antibodies need to be induced have to be very, very high titer. I mean, the adults that we studied had, titers of, 2 to the power 10 in their nose and in their serum, and some of them are still not protected. This is very, very high levels of antibody, but they're still not protected from infection. And the other problem is durability. So, whatever antibody response you might produce from a vaccine, currently, natural infection produces antibodies that only last 6 months or so. So any vaccine is going to have to overcome that durability problem if it's actually going to be useful for more than one season. And what about T cells? Well, it seems clear that it may be difficult to produce sterilizing immunity through antibody alone to RSV, so I think any successful vaccine is going to have to induce CD8 T cell responses. Ideally, the resident memory phenotypes of these T cells take up a residence in the lung and offer an early effector mechanism to eliminate virus. And these don't seem to produce enhanced disease, but clearly it's going to be very important to any vaccine that does target T cells as its effector that we don't show any safety signals or any evidence that it could contribute to immune pathology. So what strategies might we try? Well, clearly there may be a role for novel adjuvants to try and boost the immunogenicity of our existing vaccines. And other novel strategies such as viral-like particles or vector vaccines, which are currently under development, may be useful here. But I think on the whole, it's going to be very difficult to solve this problem. 40 years of quite intense research has not yet yielded us an effective RSV vaccine, despite the fact that we've got some candidates that are now in phase 2 and phase 3 trials. Most of these are just based on purified F protein, so the chances of them succeeding in producing robust and long-lasting immunity to RSV, in my opinion, is quite small, but only time will tell. All right, I've got a lot of people to acknowledge here because the sort of studies that we do here are tremendously difficult to conduct. So Chris Chu is my longtime friend and mentor throughout my whole of my academic studies, has really been an inspiration to me in terms of my own research, but also the collaborations that we've done in the work that you've seen today. Peter Openshaw is my supervisor and again, constant source of support for my work. Alan was my research nurse who did all the challenge studies with me, and immense help to me in getting all that work done. Extremely difficult. Aggie Joswick did all the flow cytometry and TISO work that you saw, and the rest of the guys here did all the bronchoscopies, so a huge amount of effort was expended there. And the remainder here is Raffiamo de Jens and John de Vincenzo, our collaborators internationally, who helped us set up the studies and do some of the TISO work, and obviously my funders. Thank you very much.